So I do want to talk about serendipity, and I will get to that in a moment. There's an old, well, not so old, maybe 50, 70-year-old blues song in the United States. Howlin' Wolf recorded it, among other people. It has a wonderful line about luck. The line in the blues song is, "If I didn't have bad luck, I wouldn't have any luck at all." We're going to talk about good luck this time. Before I actually go and talk about this, I do want to say something about one of the people that shared the prize with me, and that's Roger Chen, who died 1 week ago today. Picture of Roger. He was an absolutely spectacular scientist, and I sort of want to dedicate this talk to him. Among the many things that he did in relationship to the story I'm going to tell you is that he was the first person to come up and take green fluorescent protein and make other colors from it. He made a blue originally, and then a yellow-green, and then later was able to make through other protein colors that really went through the entire spectrum and changed it. And he did that, among other things, with a really wonderful idea in mind. And that is that he realized he could make these wonderful tools, these molecular monitors in cells, that if two fluorescent proteins were far apart, if you shone ultraviolet light on the protein, the blue protein would give off blue light, but very little else would happen. But if the two proteins were very close together, shining ultraviolet light gave mainly yellow. And because of that difference, if they're far apart, blue, close together, yellow, you can tell if most of the molecules are close together or far apart by how much blue and how much yellow are there. And what brought them close together? Calcium. And so he made a way of monitoring calcium in living cells and many other things. There's been several hundred of these monitors made. He will be sorely missed. He was really an unbelievably remarkable scientist. And I don't think serendipity really had anything to do with any of his discoveries, but the rest of us, it had a lot to do. So let me talk about serendipity and a story about GFP here. Before that, I want to just point out a couple things that serendipity, these accidental discoveries, really permeates all through the literature on Nobel Prizes. And we heard Shelley's talk, and he had a number of examples. The ones I like is John O'Keefe, who got shared the Medicine Physiology Prize in 2014. He starts off his entire story by saying this wonderful thing. He says, in the late 1960s, I was recording from somatosensory cells in the thalamus, and on one occasion inadvertently positioned the electrodes in the hippocampus. He put the electrode in the wrong place, changed his entire series of study to that, and led to his further discoveries. Already, Shelley mentioned these two gentlemen, Robert Wilson and Arno Penzias, and their discovery of the background radiation. They were doing an experiment that they wanted to use this enormous, what's called the 20-foot horn reflector at Bell Labs to, as a radio telescope, to do radio astronomy, and instead of pointing it at the Milky Way, they pointed it away from the Milky Way, where there was not supposed to be any signal at all. And they said, oh, we're going to get this really small signal, 0.7, 0.1. And they did it, and they got a much higher number, and they couldn't get rid of the number. They didn't really know exactly what was going on at first. Other people theorized, oh, you know, if there was a background radiation, it would look like this. And they said, oh, well, we have that. Got them a Nobel Prize. absolutely unexpected, probably one of the biggest discoveries in the 20th century in physics. I'm going to talk a little bit about green fluorescent protein, and it all starts with this man, Osamu Shimomura, who has a remarkable story. If you have some time, please go to the nobelprize.org website and read his story. He, at the age of 16, he's in high school. They come to his high school and they say, that's it, you have to quit school. Education is done, you got to work in a factory. Paint factory, it turns out. And that was it, school is done. He goes to the paint factory. Now the paint factory wasn't in the city he was born in. It was on the other side of the adjacent mountains in a valley. So he goes and he's in that paint factory on the other side of the mountains. That turns out to be very good because the year was 1945 and the city was Nagasaki, Japan. And by being on the other side of the mountains, he was protected when the atomic bomb destroyed the city. Couldn't go to college. There were no colleges. Everything was destroyed. Afterwards, they built the one college near him that he could go to. It was pharmacy school. He goes to pharmacy school, doesn't like it. Gets a job as a technician in a laboratory. The boss of the laboratory gives him a project, but forgets to tell him one thing. And that one thing was, I've already given this project to many people before, and everyone has failed. But I'm not telling you that. He succeeds. He's a very good biochemist. He succeeds. He discovers how a small crustacean produces light. Many different organisms produce light, and so he gets interested in that as a problem. That discovery leads him to get two things from his boss. Or two things. One, he gets invited to come to the United States. And second, his boss gives him a very unusual going away present. He gives him his PhD. So, he comes to the United States, and in the United States he's going to work on how a different organism generates light. This jellyfish. Now the jellyfish produces a green light. So he wants to know how this works biochemically. So he starts grinding up jellyfish. Eventually he starts bribing his kids to find enough jellyfish for him to grind up. And eventually he grinds them up, right? And he's an unbelievably good biochemist. Every time he does the experiment, it does not work. The whole summer, failure after failure after failure. Finally, one night, he's spent the whole day trying another experiment. It still hasn't worked. No light's been produced by any of this. He decides he's hungry. He wants to go home for dinner. And so he's a very neat, orderly person. He cleans up. And so what he does is he takes all those samples that didn't work and he throws them away in the sink. Now the sink happened to have other things in it, probably some jellyfish parts, but seawater and other things to it. It's late at night, he turns off the light, and he is about to walk out the door when he looks back at the sink. And the sink is glowing. And he looks at it and says, that's interesting. And he reasons, thinks about it a little more, he reasons, wait a minute. The seawater in the sink, it has something I've never put in the reaction. Calcium. And so he does the experiment a couple days later, and sure enough, every time he squirts in a solution of calcium, bang, he gets this flash of light. He uses that as a way of purifying the protein. He names the protein after the jellyfish. He calls it a quarin, and the quarin plus calcium gives light. But he has another problem, because he's answered the question, what's the protein that gives light? It's quarin. The problem is the jellyfish produce green light. This protein produces blue light. It's the wrong color. And so there's the second accident that happens in this research. he starts thinking about, well, how can that be? How does it get green instead of blue? And he says, there must be another protein. That protein must take on the energy from the blue light and transfer it so that it produces green light. And that's fluorescence. Fluorescence is absorbing light of one color and giving off light of a different color. And so he looks, he goes to all the samples again, but this time with a handheld ultraviolet lamp, and sure enough, there's a sample, not the one with a corn, but a different one entirely. Every time he shines the light on it, he gets this beautiful green color. And in his paper in 1962, describing the purification of a corn, footnote number 3 says, there's this other thing. If you add that other thing, you get green light. And so he called it the green protein. We call it green fluorescent protein, or GFP. And all of this together will produce the green light. I want to point out that the Nobel Prize is not necessarily given to the person with the most publications or the longest papers. This is one case where a person won the Nobel Prize for a footnote. So you can be very, very short in what you write. In any case, these accidental discoveries result in the discovery of two very interesting proteins. A corn produces light when there's calcium. So it's a calcium indicator. If you put it in cells, it's going to tell you if there's a calcium going into the cells because you're going to produce light. And as we showed, you could look at proteins, or you could look at GFP, shine blue light on GFP, and then get green light coming out. And I heard about this in a seminar that was in my department in 1989. So we're going from 1962 to 1989, and I'm listening to a seminar, and it was a seminar that I had absolutely no interest in at all. It was just, well, I may as well go there, everybody else is going there. And I'm sitting, and I hear about this protein. And the protein will give you green light if you shine blue light on it. And I realize it's a lantern. All that you have to do, it'll show you wherever it is by just shining blue light on it. And I get very excited, but I want to tell you why I got very excited. At the time in my lab, I was working with this small animal. Craig Mello will also probably talk about this. This is a nematode, a roundworm called Serendipity elegans. And I had been working on this for about 12 years when I heard that seminar. Now, in those 12 years, I gave many talks. And every talk I gave, I would bore everyone to tears saying why this is the best thing to work on. And one of the things I would always say is this animal is transparent. You can look right through it. You can see every cell. After 12 years, I even believed that. So that's one thing I brought to that seminar, the fact that I worked on a transparent animal. I didn't work on mice or chickens or fruit flies or whatever. I worked on a transparent animal. At the same time, we were doing studies to understand how nerve cells were developing and functioning. And I don't have time to go into all of that. But suffice it to say that we had started cloning genes that were needed for a small set of nerve cells to function properly. Now, when you clone a gene, one of the very first questions we asked was, where is that gene active? And at the time, there were many ways of doing this. We could, because DNA makes RNA, makes protein, we could make an antibody against the protein and see where the protein was made. Or we could use a method called in situ hybridization, which tells us where the RNA is in the cell. So we could see where the RNA was made. Or we could use what are called reporter constructs, one being the gene LAC-Z from E. coli that makes the antibody, the enzyme beta-galactosidase, and the blue indicates where that enzyme is. So any gene that turns the making on of the enzyme, we could see it. It reported where it was active. So I was interested in I was working on a transparent animal, and I wanted to know where genes were turned on. And I go to this seminar, and fortunately, the seminar, the part that I told you about Shimamura, that was in the first 5 minutes of the seminar, that is before I fell asleep. So I was listening to that, and I was saying, wait a minute, I work on a transparent animal, and I want to know where genes are active. I could use this, and I could see it in a way that we couldn't with any of these. The problem with all of these methods, they all worked, they all gave us the answer. So we weren't going after, how could we do this experiment? We had answers, we knew what cells this work did. But every one of these methods required us to take the animals and do a lot of preparation. We had to fix the animals, that is, make sure nothing moved. We had to permeabilize the animals, making sure that the antibody got in or the reagents got in. We were looking at killed samples. Nothing changed. If we wanted to look at what happened over time, we would have to take the animals and prepare it at this time, and then this, and then that. But we never really saw something happening over time. And that's what life is, looking at dynamic, not static situations. And I realized, wait, if we put GFP in here, it turns on, we can watch this in a living animal. And that got me very excited. So, this is what we did. I should say something about my color code. All the people you're going to see pictures of, they all have their names in different colors. Anyone in my lab, their name is in red. Anyone that is a collaborator, their name is in blue. And anyone whose name is in black did something I wish I had done. So, Douglas Prasher was isolating the DNA. We had a new student in the lab, Gia Skirkin, and that was her project. See if you can put green fluorescent protein into bacteria. 1 month after she started graduate school, bang, she has a picture. She had green fluorescent protein in this. This was a wonderful, wonderful result. We did a lot of jumping around in the lab when we had this. And it was one of those rare cases when the experiment works the first time. I want to point out one other thing about this page from her lab notebook. This is the way, I'm trying to tell you that the standard way of doing experiments is not what happens, right? You throw things in the sink. By the way, that's a very tried and true biochemical procedure. If your experiments aren't working, throw it on the floor, the desktop, or the bench top, or the sink, and then do the experiment. It often works. In any case, on this page, there's this part I'm going to circle up at the top there. And what it says is that in order to see this wonderful picture, take this wonderful picture, Gia had to go to another lab that had a better microscope, because our microscope was a piece of junk. As a result, when she made this wonderful finding, we couldn't do any more experiments because they wouldn't let us use their microscope anymore. I solved that problem by calling up all the sales representatives in New York City and saying, we have just discovered a new way to see where genes are active. And it uses a fluorescent microscope, and I gotta tell you, we gotta buy a new one. But I don't know which one I wanna buy yet. So if you can bring your microscope to my lab and let us try it out, maybe for 1 or 2 months, then we're going to decide what to do. All the experiments we did were on borrowed microscopes. In any case, they worked, but I want to tell you how strange that was. Because people knew about green fluorescent protein before we did the experiment. And if you asked anyone who knew about green fluorescent protein, can you put this into bacteria and they'll turn green? They would have said, no, you stupid person, it's not going to be possible. And the reason it wasn't possible is something so strange happens to the molecule. No one knew how that came about. And what they found was the following. You know that proteins are long chains of amino acids. Each is connected to another, so you could find a chain of atoms that go right through the molecule called the peptide backbone. But GFP was incredibly strange. Instead of having this line of atoms, the mature protein had the line interrupted by a circle. Right here, it changed into a 5-membered ring. And no one knew how this took place. They thought, well, maybe there's an enzyme that converts this, or maybe 2 or 10. But it can't do it on its own, so don't even try the experiment. So we tried the experiment, and it worked. That says there is no other enzyme. It can work completely on its own. Now, I want to tell you how lucky we were. This is 1992 when we're doing the experiments. Now, in 1992, there were 2 ways of doing experiments where you wanted to get a large amount of DNA. If you were careful, you had something reliably make the DNA, like bacteria or a bacterial phage. You put it in the bacterium and let it grow, all the bacteria grow up, or all the phage grow up, and isolate the DNA. Then you'd use one of Rich's restriction enzymes, and you'd cut it. And that you'd get this wonderful piece of DNA that was basically perfect over and over and over again. The other way was to use enzymes that amplified the DNA by polymerase chain reaction, but they were notoriously bad. They would introduce mistakes. So if you wanted to be careful, you used the restriction enzymes. And this turned out to be very important. Here's a diagram of the piece of DNA we were trying to copy. The green part is the part that encodes for green fluorescent protein. The red part is some other strange jellyfish DNA. We didn't know what it was. There were 3 other groups that tried the experiment. The ones that were careful, the other 3 groups, they said, we want the DNA to be perfect. So they grew it up, and they cut it out with the enzyme EcoR1. But that piece will have the red bits and the green bits. And then they did their experiments. I didn't want anything extra, and I said, look, we're putting this in bacteria. We don't care if every bacterium is green. We just want at least more than one. So if 90% of the stuff is bad, I don't care. 10% will be good, and we'll be able to see it. So let's only amplify and just get the green bit. And that's what we did. So you amplified the green bit, put it into bacteria. It worked great. Not every cell, but enough so that we could see it worked the first time. The other 3 groups, the ones that were careful, they said, we tried it, doesn't work. Now remember, if it doesn't work, there must be that converting enzyme. There must be another thing there, because that's what went wrong. So they were careful, and it failed. We were sloppy, and it worked. I'm going to come back to that idea at the end. I'm going to give you some ideas of how to increase serendipity. In any case, we eventually did publish this. I'm going to skip this because I know we have other people we want to talk with, and there we go. Along the way, other people tried it. This person did some wonderful work showing that she could get GFP attached to a protein, and that protein, wherever it went, would take GFP with it, and she could follow it. And I wanted to cite her work in our paper, and she made a lot of demands before we could cite her research. She said that I had to make coffee every Saturday for 2 months. I had to prepare a special French dinner for her, and I had to take out the garbage nightly for a month. This is my wife. I will also tell you that this is the only part of my talk you will remember. In any case, what she did was this wonderful thing of following a protein by having GFP attached to it, the lantern attached to the protein. I'm going to show you some other pictures of GFP or some other things. It was wonderful because it could be inherited. You just give the DNA to the organism. Using it didn't cause a lot of problems. All you had to do is shine blue light. You got green coming out. It filled up the cells. It could be seen in living cells and living organisms. Here's a set of organisms with it. Our worms. This is all the nerve cells. Here's a worm, fruit fly, mice, zebrafish, canola plant. This is Alva, the GFP bunny. That was commissioned by a Brazilian artist living in the United States to show its art shows, to show the connection between art and science, and some cells here. I want to show you some cells dividing. These are nuclei dividing in Drosophila, and the one on the left here, GFP has been attached to a protein of the spindle. You see the spindle forming in cell division. There again, it's very much sped up, but you watch this happen. And I hope that you all have this one question that comes to mind by looking at this. Why are they all doing it at the same time? What synchronizes this? And in fact, we know quite a lot about what synchronizes this. Over here on the right-hand side is a falsely colored picture. This picture is, again, of the nuclei dividing, but the colors are, if you have very little, it's blue, a little more, it's green, a little more, it's orange, a little more, it's red. So this is still GFP, but different things. And we're going to see this now, going, hopefully. So the nuclei break down, now it's all over the cells, because this is GFP attached to a small peptide that brings it into the nucleus, if there is a nucleus. But once the nucleus breaks down, it goes all over the embryo. And what you should see in this picture is it's not synchronous. It's going as a wave from the bottom right to the upper left. And that says, but wait a minute, it's the same animal. Why is that not synchronous, or this sample? Why is that not, why is it going as a wave? It turns out, this is a very subtle example of damage. The cover slip is a little too tight at one end, and so they've damaged the embryo, and that's why this pattern comes up. I've already mentioned Roger introduced all these wonderful colors and stuff. This was used by Jeff Lichtman and Josh Sains to develop this wonderful way of marking cells that they use for the mouse brain. And if you use every imaginable color, right? 4 colors in random combinations. If you get every imaginable color in the spectrum, and you do this in the central nervous system, in the brain of an animal. There's only one name you could use for this, and this is what they called it, and that is brainbow. Really magnificent pictures here. GFP has been used in many ways. I'm going to skip some of the things here, fundamental research, applied research, and some really unexpected things. People have tried to use it to detect landmines. They've made silk clothing by putting GFP and RFP into silk moths and then making the silk from them. I'm not sure why one would want to have fluorescent clothing, because you have to bring an ultraviolet lamp with you all the time as you're walking around, but it was still interesting. And some of you may know that there actually has been, at least according to this source, a transgenic human with GFP in him, and it explains a particular property of him, and that's this individual. I can tell you the story of that later, but I don't want to go into this. What I really want to say is that I hope you've gotten from my talk and Shelley's talk as well, the idea that many, if not most, discoveries are accidental. So if they're accidental, how can you sort of up the amount of accidents that happen? How can you improve serendipity? Where does it come from? Now, it's not just having accidents, because you really have to think about what you got that was strange. But I have 4 ideas about how to increase serendipity. The first thing is to question and to wonder. To always be asking of the results, does this make sense? Can I understand this? If not, should I be thinking about something else? Always question the ideas. There are so many things around us that we never, ever question. I'll give you one of my favorites. And I'm sure this is something that everybody in this room has experience with, but I'm not sure that everyone in the room has thought about this. When you have chicken or turkey, you know that there's white meat and dark meat. I don't know how many people know why there's dark meat and white meat and what the difference is. The dark meat is muscle called slow twitch that needs to work constantly. It needs to get oxygen, and it has a molecule in it called myoglobin. And it's the myoglobin that makes it a dark meat. The breast meat in a chicken or a turkey, that only has to work a little bit. It can go into oxygen debt. There is no myoglobin, and as a result, it's white. So the difference between white and dark is the difference of the 2 types of muscles and 1 protein that's in there. The next thing is, if you want to have the accidental discovery, you got to do a lot of experiments. Right? If you don't do a lot of experiments, you don't learn a lot. You don't have the unexpected arrive. The famous ice hockey player, Wayne Gretzky, has a wonderful line. And he says, you're going to miss 100% of the shots you don't take. You got to take a lot of shots. Every once in a while, the accidents come. The third thing that I want to suggest here is you should do controls. In people working on the worms, one of the famous control experiments was done by a terrific scientist named Cynthia Kenyon. Cynthia Kenyon got interested in what made the worms, some worms, live longer than others. And she was working on this and trying to do the studies, and she said, I need a control for this. I have the wild type animals, but what should I use as my control? And she says, I know, there's an animal I'm pretty sure doesn't get older. It ages at the same rate. It's not going to be, I'll use that as my control. And so she tested the wild type, and then she tested the mutant, and the mutant lived twice as long as the wild type. And so all of a sudden her control became her experimental, and she started discovering genes that actually, when those genes were knocked out, the animals lived longer, and has done spectacular work on that ever since. And the final thing gets back to that experiment I said about sloppiness. This came up, and so this is, don't over-control your experiment. Do the controls, but don't over-control. And this idea actually I got from a wonderful interview with one of the great scientists of the 20th century, Seymour Benzer. And in this thing, he was actually talking about one of the people that was his mentor, the Nobel Prize winner, Max Delbruck. And he was talking about them, and he said Max Delbruck had a theory about how you should do experiments. And this theory was called the principle of limited sloppiness. Now, you didn't want to have everything be exceptionally sloppy, but you also didn't want to control absolutely everything. And this came up, according to Benzer, in a wonderful experiment that people were trying to grow bacteriophage on Petri dishes. And what they would do, to show you pictures, they stacked the Petri dishes. So the light was up here. And you can see this is lighter than this. It's very dark down there. And they grew up the phage, and they realized that every time they did this experiment, the plates at the top had much more phage than the plates at the bottom. And they discovered light induction of the phage, something they would never have done if they controlled the experiment by putting all the plates on the same level with the same amount of illumination and everything controlled. So this was limited sloppiness. So sloppiness is okay. It's probably a good thing. Thank you very much.